Good morning. Well, thank you very much, first of all, for this opportunity to present. I'm delighted to be here. This is my first Barth syndrome meeting. What I'd like to do for this presentation is first give you a quick overview on why am I here? Why do I conduct the research that I'm doing? And then just go through some of the genetics, genomics, some of the data that we have, and which really focuses on the family. And why am I here? So when I see patients and families with cardiomyopathies, what I'm very intrigued by, the variability. We see this in many different genetic disorders, not only Barth syndrome, but why with the same mutation that segregates within a family or between families, why is there such clinical variability in that age of onset, severity? You'll see different families with cardiac disease, severity, heart transplant. So not only the primary mutation, so when we identify a primary mutation in a family, it's often very difficult to get prognosis because of this variability. We tell them what to prepare for, but we really, to get to personalized medicines, we really have to understand the variation and the mechanisms behind that. So the goals in my laboratory is not only to look at the primary mutation, for example, tafazin mutations, but what is behind this variability, the secondary variability, and certainly the sequence alone is not going to tell you everything. You have to develop very good models, for example, what we heard this morning. So the family I'd like to discuss is a family which we presented a couple years ago, this is a 53-year-old gentleman with features of Barth syndrome and a tafazin mutation. He is currently the oldest individual with a tafazin mutation published. And he, as a child, he did not have any cardiac disease, lived on a farm, he worked, had a chronic myopathy, but was able to function and do fine until his late 40s when he became fatigued, which led to a cardiac workup. And at age 45, he had his first echocardiogram, and they found non-compaction. Okay? Now, 2 years after his diagnosis, his great-nephew was born, and he had the classic typical features of Barth syndrome: neutropenia, 3-methylgluconic aciduria. He had a dilated cardiomyopathy, and then unfortunately had heart failure and had a transplant as an infant. So this is a family we reported. Many of the clinical details, please go to this particular publication, but you could see in this family this kind of spectrum of variability. Now, genetic modifiers, we talk about it. You hear about this in the meeting. You talk about the families, but how are we going to understand this? So what we have done was to take a genomic approach to see if we could start to understand genetic modifiers. And again, this is the tafazin mutation that both the proband and the great-nephew has. This is an exon 7 non-sex mutation, so a truncation mutation that you see in normal individuals. Here's the homozygous mutation in the affected males, and then the carrier females over here. This is in the publication. So the real question is, why in families with the exact same mutation, why is there such clinical variability? So it's a very exciting time in genetics genomics because now, with a new technology, we can get away from the single gene analysis, like I showed you, the Sanger sequencing, and actually look at a catalog the variation in all the genes, the 20,000 genes. So the simple idea is that if you just look at allele sharing, so there are about third-degree relatives, so they share, at least by identity, by descent, about 1/8 of their genome. Over here would be the ones which are shared, including the tafazin. And the simple hypothesis would that perhaps the older individual with less severe symptoms would have some sort of suppressors. That is, their system, a combination of variants, their genomic background has been able to suppress the deleterious effects of tafazin. Whereas in this case, perhaps, there are these enhancer variants, or this enhancer genome combination of variants that brings out the features of the condition. So what we did was, again, this is just simply, again, reiterating that on just a single gene model. But certainly it doesn't have to be a single variant that's doing this. It could be a combination or actually the entire genome. Whereas the suppressor variant were analogous to give you the clinical variability. So what we did was we did exome sequencing, and I won't go through the detail. We took the genomic DNA from the 2 affected individuals, did enrichment to enrich for variants in coding regions, 20,000 different genes, including tafazin, did a lot of bioinformatic analysis, so this becomes more a bioinformatics analysis after this to look at the data. And if you look at the data for the exome sequencing, and I was born and raised on Sanger sequencing, so every time I look at data like this, I still have to scratch my head and said, this incredible. So the raw sequencing data in the proband here is 13 gigabases. So the Sanger sequencing data on a good run, you can get 300 to maybe 800 base pairs on a single read, nice and clean. But here you can see 13 gigabases, 14 gigabases on the nephew, and the number of reads, so 138 million reads. So not one read like the Sanger, you're getting multiple reads. So this is the power of the next generation sequencing. So when you map that back to the genome reference database, you can see most of them map back. And out of the 200,000 exons of the 20,000 genes, you get a pretty good pickup rate of more than 90%. So one thing, the sequencing is very good, but it's not 100%. Okay, so the data is still incomplete, but this is reasonable data to look at the variants. So if you took that data and you just look for variant alleles compared to the reference sequence, you could see a large number, proband over 100,000 variants, great nephew over 100,000 as well, and then you have to, what we did was filter them. Okay, because this is the problem. I speak with systems biologists, genomics. How do you analyze multiple variants? We really don't have a good method of that yet. So what we did was use one approach. Question was, well, if I was a modifier, what would I look like? Well, I don't know. Would I be a common variant? Would I be a deleterious variant? I don't know because we don't have any information about that. So using the same principles to identify a mutation, that's something that's predicted to cause an impact on the gene, the protein, and a functional effect, this is the approach we use. So if you look at these variants and filter them by those which are predicted to be deleterious. So these are ones, the changes that fall in genes. They fall in the coding region. And are predicted to be either nonsense mutations, truncation mutations, indels or frameshifts, or missense. So you can see a vast majority of these. One, there's a large deleterious mutation burden. Now don't be surprised by this, because if we sequence everyone in this room, we will have very similar data. Okay? You will pick up individuals, population data shows that you will pick up a large number of nonsense and truncation mutations in different genes. The problem is trying to figure out if these mean anything. Are these primary mutations that cause disease, like Barth syndrome? Are these modifiers with the combination? We don't know. But so this is not surprising that you see this many. So what we did was then see, going back to the sharing, let's see how much these are shared. So roughly about 2/3 of them are shared between the individuals. Now that's more than 1/8, and that's because the ancestries of these two individuals are very, very similar. The closer the ancestries, the more common the variants are going to be. But you do see about 1/3 of the variants only found in the proband, and only 1/3 of the variants found in the great nephew. Okay, so these are the collection of variants that would make very interesting potential candidate modifiers. So we focus on these. Now, 3,000 is still too much to analyze, and this is just to show you some of the next generation sequencing data. So here's the tafazin gene over here. The red is the variant over here. Each one of these are the single reads. So you could see it's hemizygous for the mutation, and again, Sanger sequencing, if you want to see that. Now, how do you look at those 3,000 genes to pick out potential suppressors? What we did was look at, well, why don't we focus on those genes that have been implicated in tafazin function, okay? So looking at different pathways, we focus on, of the 20,000 genes, 264 genes, so this includes about 100 genes involved in phospholipid metabolism, 3-methylgluconic aciduria, what pathways are implicated there, as well as a large collection of genes involved in muscle contraction and dilated cardiomyopathy. Just to give us a focus, because those are the ones that we could target and do functional studies and hopefully prove that these modifiers are actually doing something. So when you do that kind of filtering, the numbers reduce to close to 100, and you could see 72 variants are shared, but then you still have a collection of variants which are potentially unshared and be potential modifiers. So now, my experience with exome sequencing, it's a very good technique, but still, because of the nature of the technology, there are many false positives, so we went through those collections and Sanger validated them, okay? So these are real variants. And what I'm going to show you is quite raw. That's the raw meat here. That these are potential. Please don't take away with these that we've identified these modifiers. These are still potential candidates that need to be tested. But these are rare alleles, so they are less than 1% of the population. These are based on the exome sequencing databases of thousands of individuals. These are at sites which are highly conserved, very highly conserved. So these are predicted and associated with tafazin function. So predicted to perhaps cause a change. And I'm not going to go through each and I'll go through the tables because these are just the Sanger validation of the proband nephew, just to prove that these are real variants. And the first set are those which are found only in the proband and not in the great nephew. And these are our list of potential suppressors right here. And just to go into a little bit more about what these do. So these, again, are heterozygous, rare variants, highly conserved, tafazin-associated function. There's two particular genes over here which are involved in metabolism. These are mitochondrial proteins, so they make very, very interesting candidate suppressors. Now, yesterday, my poster, Dr. Kuchua, pointed out this particular candidate right here is very interested, and led me to have a discussion with Dr. Hatch. Because as you heard from the first presentation in this session, and there's so many different names for these genes, this particular gene is the same gene that they've introduced in the Barth syndrome genes that rescued the function. So this is the MLCA, it's that same locus, for example. So that's very, very interesting that in this particular family that we did identify as a potential suppressor of the tafazin mutation, the same protein that's implicated in rescuing tafazin mutations, restoring cardiolipin levels, okay? Now that's a very interesting that needs to be followed up. So we'll do functional studies to look at this. It is heterozygous, and so it's interesting to see whether or not there is perhaps increased activity of this particular protein that might be compensating for the defect in tafazin. Now, for each of these, I can make a case that these are very good suppressors. This is a gene, mitochondrial, that's involved in protection against oxidative stress. Titin is a very large protein of the sarcomere. Truncating mutations are found in, it's estimated about 25% of patients with dilated cardiomyopathy. Okay. So is this a second mutation? Is this a modifier? What is this doing in terms of sarcomeric function? Titin is a protein that's involved in elasticity of the cardiac muscle. So is there modifications based on one or two of the variants that protects the individual against heart failure? I'm not really sure. Now, likewise, we validated the potential enhancers, and the short list is over here, and those are three that we found here. So these are the ones that are found in the great nephew, but not in the proband. And here's a list for here. And again, I can make a good case for each of these being very good enhancers. Certainly, ryanodine-2 is involved in calcium homeostasis, TGF-beta, and signaling, and again, another rare variant that we found in Titin. So where do we go from these? Because this is just a start. I think we're doing a very good job in understanding the primary mutation. We have the tools to look at that. We can identify the families. But I think what's happening in genetics and genomics is really trying to shift and trying to understand this as a system. What is the collection of variants on the system? how come you have one mutation that has a defect? Why is that variable within families? So this is one approach. So what we're doing is moving to the next step to do functional models, because you need to test these variants. You're picking up all these variants, like the Titin mutation. Is that a primary mutation? And then so you can look at different combinations. And then so we've collected fibroblasts from the proband, and we are working towards developing a stem cell model as similar as you heard. So hopefully that we could show quantitatively on a cellular basis that there is a difference in severity in the cellular phenotype, and then quantitate that. And with those suppressors, if those are really suppressors, you should be able to introduce those into cells of someone who's more severe and suppress the phenotype. Likewise, if those are truly enhancers, take those and move those into cells of the less severe and bring out the phenotype, the cellular phenotype. So those are long-range goals, and hopefully at the next meeting I'll have some functional data to present in terms of these modifiers. So again, I'd like to thank you for your attention and the individuals in my group, and thank you very much.
Thank you. That was educational and frightening at the same time, I think. Do you want to go? Yeah.
Great talk. Can I have the previous? Yeah, there's a pedigree here. It catch my eye is the proband, the 53-year-old male that actually have children. So you actually determine that's his children.
So these are adopted. You can't see the brackets.
Okay, so one is adopted.
He does have one, right, and he's unaffected.
The other son, you predict the son will not be affected.
Exactly. Right.
Okay.
I don't know if he has that Titin mutation or the Titin truncation.
As you know, a long time ago when we have the fly models, the fly models of Barth is male sterile. And so at that point, we sort of raised this issue whether the Barth men are sterile or not. And but now because more patients become become the age that could have families, and so that could be a issue now. And I'm not sure anybody look at the mouse models whether the male there is sterile, because we have knockdown mouse. Maybe it's not exactly the ideal for testing that. The knockout mouse model for Barth syndrome maybe more suitable for testing the sterility issue.
Thank you.
I have one question. Do you have performed a cardiolipin profile and do no? Okay,
we have not done
because it should maybe look like the family of Collins from yesterday. Maybe.
We'll do that on the fibroblasts.
Just one curiosity. The first family I saw, the grandfather was affected and had 5 daughters, all of whom were carriers. And his grandson, whom I follow, has a child too. So fertility is pretty good. It's interesting that the Titin mutations are common. The truncating one's not as common, but we have essentially identical story. An adult who was asymptomatic throughout childhood, who was picked up with LVNC and some symptoms, some dilated cardiomyopathy in his 20s, and he's got a Titin mutation as well. So it's because it's a common gene that's mutated. It's hard to say, but it's the fact that it was the same story.
Exactly.
Makes you wonder.
Titin's quite interesting because you can get primary mutations that look like real mutations segregating with the phenotype, but then since it's such a large gene, for example, we sequence everyone here, we'll pick up missense and probably some individuals with truncating heterozygous Titin mutations. So maybe Titin, because it's so frequently in the population to have these deleterious mutations, maybe in combination with other cardiomyopathy-causing mutations like TAS, because I have another family, some of these other families, if you notice by the pedigrees, are autosomal dominant. They have mutations in lamin AC. And we also have a very interesting hypothesis where we with Titin. There's families described in the literature with combination of Titin and lamin AC mutations and might bring out the phenotype more. So Titin still is a very interesting candidate gene. If we could look at that functionally to see what's the combination alone and with the Titin and the tafazin and together, you know, that'd be a very interesting story. Perhaps Titin, you know, is still an interesting candidate.
A very interesting talk. I was wondering, when you did this next gene sequencing, did you get any mitochondrial DNA genes?
You know, we sequence mitochondrial DNA separately. And as you'd expect, it's the same with all the individuals since it's maternally inherited, because another hypothesis would be mitochondrial DNA mutations giving the variability. I think that would be interesting if we look for heteroplasmy in cardiac tissue. You know, germline, there's the mitochondrial haplotype is the same, you know, along with, since it's maternally inherited, it's the same. And there's no additional mutations. We sequence both these individuals to the exact same mitochondrial haplogroup, and there's no heteroplasmy, at least in blood and saliva. You know, but if we look at heart, maybe there's a second hit in the mitochondrial DNA as a modifier. Okay.
And the left ventricular non-compaction, was it in both patients?
Well,
or only was one?
It was primarily described in the proband, but when you look at the echocardiogram, there is some hypertrabeculation, but it's dilated with hypertrabeculation and then heart failure. So again, you go through the different the variability within non-compaction.
Maybe it will be a good idea. Yesterday, Lynn Jeffries showed this list of the genes which are also implicated in the left ventricular non-compaction, and maybe it will be good to also to pick up something from there.
Absolutely, and many of those genes are in the cardiac function dilated cardiomyopathy list. There's about over 100 genes implicated in different cardiomyopathies, and those were included in the filtering, right?
Can I just ask you, you said there's truncation mutations in 25% of DCM patients in Titin?
Yes.
What, is that the same in normals as 25%?
No, actually, it's from the Seidman group. They looked at over 200 patients with dilated cardiomyopathy. It's a New England Journal of Medicine paper, and about 25% have truncating mutations. In a cohort similar size of hypertrophic cardiomyopathy and controls, it's much less. So it's significantly elevated. Now, what we have, some of those did not segregate. Those in which it was familial, not all of those segregated with the phenotype. You know, so again, is it a polymorphism that's not doing anything? Is it a modifier, or is it a primary mutation? We don't know. But still, the thought is Titin has been implicated in some way to contribute to dilated cardiomyopathy.
Very interesting. We'll make this the last question.
Michael, I forgot to ask you at your poster yesterday, but do any of these variants, are they predictive of any change in structure of the proteins at all?
You know, I'm just going through that. You know, certainly I was focusing on Titin until we had our discussion. Many of the Titin ones are the ones on the C-terminus that changes the interaction with the thick filament of the sarcomere. Now the other ones, like for example for the enzymatic ones, I'd have to look. Yeah.
Okay, thanks for educating us all. That was superb. Thank you.